We will now talk about catecholamines. Welcome to our channel, Internal Medicine Made Easy, the online learning platform that provides medical education to healthcare professionals and students to improve their clinical skill and help them in their studies, courses, careers, and research. Organ systems involved: neuroendocrine chromaffin cells responsible for the biosynthesis of catecholamines are located throughout the brain and in the adrenal glands. Biosynthesis: catecholamine synthesis within the adrenal medulla is controlled by serum concentration of the amino acid tyrosine. Tyrosine undergoes hydroxylation via tyrosine hydroxylase to form DOPA, which then undergoes decarboxylation to dopamine. Dopamine may be secreted into the bloodstream or undergo further hydroxylation to norepinephrine. Norepinephrine can be secreted into the bloodstream or further modified by methyltransferase to epinephrine. Mechanism: after an external stimulus triggers the body stress response, the sympathetic division of the autonomic nervous system is activated and acetylcholine is released from sympathetic splanchnic nerves. Acetylcholine binds to nicotinic receptors located on the membrane of chromaffin cells in the adrenal medulla. These receptors promote exocytosis of catecholamine-filled vesicles for transport in the bloodstream. In the blood, catecholamines target alpha and beta adrenergic receptors. Function: the fight or flight response of the sympathetic nervous system is a direct result of the multisystem action of catecholamines. Secretion from the adrenal medulla regulates blood pressure by contracting the smooth muscle in the vasculature via alpha-1 receptors. Further musculoskeletal actions of catecholamines include enhanced contractility of cardiac muscle via beta-1 receptors, contraction of the pupillary dilator via alpha-1 receptors, piloerection via alpha-1 receptors, and relaxation of smooth muscle in the gastrointestinal tract, urinary tract, and bronchioles via beta-2 receptors. Both epinephrine and norepinephrine modulate metabolism to increase blood glucose levels by stimulating glycogenolysis in the liver, increased glucagon secretion, and decreased insulin secretion from the pancreas, and lipolysis in adipose tissue.